Thank you, Ada, and thank you, everyone. It's a great honor to be here in person. This is my first time in person, and thank you for staying. It's a long day, and chemistry is at the end, but I'll try to keep it a little bit lively. It turns out I'm a scientist and an engineer who spent her career observing and manipulating the evolution of proteins, breeding them in the laboratory like you breed cats and dogs so that they exhibit new properties, new traits. The scientist part of me is delighted by what I've learned from this and what I can create using this powerful design process of evolution. And the engineer in me pushes me to implement that new knowledge and insight into how biology innovates in order to solve problems for people. So I've worked on everything from converting biomass to jet fuel, to replacing toxic insecticides with sex pheromones. And I've explored and exploited the power of evolution with an eye to helping us live sustainably on this planet so that we can share it with people and with the other species on which we depend. So nature is surely the best chemist of all time. And her enzymes, these are the catalysts of life, are responsible for all the marvelous and ingenious chemistry of the biological world. Yet I still want to make new versions of that, things that are useful to human beings, and I'll talk more about that later. But I also want to explore new chemistry nature never exploited as far as we know, and chemistry that humans need and perhaps even invented, or chemistry that remains to be invented. Now, what an enzyme can do, that is, its function, is determined by its amino acid sequence. So Demis showed us that the amino acid sequence determines its structure. I'm not interested in its structure. I'm interested in what does it do? And what it does is a big step beyond. And it's probably an even more complex problem. So Demis hasn't solved that one yet, but maybe next month. I figure next month. But I'll tell you, it's really important to do this because what a protein does is what matters in the real world. Not a game, right? What it does is what we need and what matters. So that's the problem that has to be solved. Now, the tools of molecular biology are really remarkable. As you know, we can read DNA. Sequencing a human genome costs a few $100s. The progress has been amazing. We can write DNA. You can type out a sequence, email it off to a supplier, and you get the physical gene back in the mail. We couldn't even think of this 20 years ago. We can edit DNA. Nobel Prize to Emmanuel Charpentier and Jennifer Doudna. That means you can go in and make modifications to DNA inside of a living cell. But what we struggle to do, and what I work on, is composing it. So we can read, we can write, we can edit DNA, but we don't know how to compose it, even a single enzyme. So to me, this code of life is like a Beethoven symphony. It's intricate, it's beautiful. It's the product of evolution over billions of years. And we humans don't know how to write like that. So to sidestep this profound ignorance of how the sequence encodes the function, I turn to evolution, a simple process, indeed an algorithm, right? A simple mutation and natural selection that over eons of evolution has given rise to all the diversity of the biological world. Just look outside or look at the most complicated things in this room, the human beings. These are the products of this simple algorithmic process. And there's no engineering process like this in any human endeavors that works from molecules to ecosystems. It's really quite amazing. So let me first ask, though, where could we go if we could evolve? So this is also picking up on what Demis said. There's a universe of possibilities out there, possible sequences that nature never had a chance or even a reason to explore. So nature's proteins from the beginning of time have explored the tiniest fraction of this universe of possibilities. That's what biologists study. I, as an engineer, can consider this entire space. And I'll tell you, out there is the solution to the energy crisis, the cure to cancer, maybe even the cure to death and taxes, or at least taxes, if not death. And it may interest you to ponder this space. It's even bigger than the space of possible conformations, bigger than the number of particles in the universe by many, many orders of magnitude. And how does one discover a useful protein in this infinitude of possibilities? And I liken this, I use Jorge Luis Borges' fabulous short story, The Library of Babel, which I'm sure all of you have read, to describe this space, because in this fabulous short story, he describes a library of all the possible books you could assemble by randomly permuting the letters of the alphabet in 500 pages. And of course, most of those texts are gibberish, right? They don't encode anything meaningful, and his despairing librarians throw themselves off the balconies in despair of ever finding a single meaningful sentence, much less a complete story. So if we're trying to evolve in this vast space, how does one find meaning? How does one find something better than what you can scrape off the bottom of your shoe, the product of evolution? So similarly, most proteins encode nothing we would recognize as meaningful, but I am nonetheless surrounded by meaningful stories on the bottom of my shoe, in the air that I breathe, or extracted from a database. And these are the products of work performed by mutation and natural selection. And evolution continues to create new ones. So I decided to start my own exploration by using this gift, the products of billions of years of evolution, as well as this process. So the challenge then is to discover new sequences, deliver improvements on a scale of weeks rather than millennia, or rather than 1 billion graduate students. And so many years ago now, I used emerging tools, difficult tools of molecular biology, to practice evolution in the test tube, exploring new enzymes, taking the simplest possible search strategy of making random mutations at a low level and screening, just sorting through those for desired changes, much like you breed cats and dogs. So by doing this, we could nudge enzymes over just a few generations into whole new niches, whole new functions. And so lots of people have used that approach to make everything from your enzymes in your laundry detergent. Go back home and you'll find that those are your laundry detergent is full of enzymes. All of those are optimized by directed evolution. Reagents for DNA sequencing, enzymes used in reducing pollution from livestock, used to manufacture everything from paper to pharmaceuticals. Now, more exciting than that, though, to me, is that we can use evolution not just to improve, right, but to improve on the chemistry that nature has already discovered, but also to really innovate, sort of the equivalent of creating new species. And when I say innovate, that means create enzymes for entirely new chemistry, not known in the biological world. And also, if you think about it, nature has done that countless times. Because if we started from a last universal common ancestor, there had to be a lot of inventions over the period of evolution. So I ask, why not use this powerful process to explore the future? Evolution hasn't stopped. It's going into the future, and we can actually explore whole new futures this way. So I think I have time to share just one fun but interesting example, and that's constructing carbon-silicon bonds. You can look everywhere you want in the databases, but you never found an enzyme that could construct carbon-silicon bonds. And as far as we know, nature does not do that. Humans do. There's 50 products in this room, and we make billions of tons of things with carbon-silicon bonds. your earbuds, your hair gels, the caulks, the sealants, the paints. There's a whole huge industry that makes organosilicon compounds using expensive platinum catalysts whose mining tremendously degrades our environment. But biology doesn't do that. So starting with a simple chemical hypothesis, we tested very simple proteins containing an iron atom in a heme, and it's too late for any chemistry, so you don't have to look at the chemistry, but notice at the bottom, there actually is a silicon-carbon bond in the product that wasn't there to begin with. We discovered that a protein from a hot, salty pool pulled out of Iceland, but we pulled it out of a database, catalyzed a simple carbene transfer reaction to make a new carbon-silicon bond. And furthermore, it's not its natural function, but it evolves. And we use this process of directed evolution, make mutations and screen for better function, to make the best version of this catalyst that a human being has ever seen. And so it does it much more precisely and much more efficiently than any human-made catalyst. And I present this example because it beautifully illustrates how nature innovates, right? Basically, it uses the diversity that's already there. There was this protein sitting out in a hot, salty pool that didn't know it could make carbon-silicon bonds until we came along and asked it. And when we asked it, we found out not only did it do this, but then it could get better and better at it. So this shows you that nature's diversity is what fuels future evolution and fuels the discovery of chemical innovation. So this demonstration that biology can make such bonds delighted us, and it also delighted the world, especially with the help of journalists who, you can see Spock and Captain Kirk there looking for life in rocks. We said nothing about putting life in rocks. In fact, we never said anything about silicon-based life, but the whole world just took this story and ran with it, and I think 12 people actually read our paper. But it was something that people wondered about. And the views on the paper divided up among those who felt that silicon-based life was something possible and those who didn't. So we had lots of criticism and lots of support, and it had nothing to do with what was in the actual publication. But what I hope you'll take away from this short talk is the wonder of this process of evolution, because it will continue to innovate. In our rapidly changing world, we're providing new niches, new opportunities, new toxins, new plastics on which evolution can discover new capabilities, and we can explore possible futures in the laboratory. Now, in my last couple of minutes, I want to share this vision of the future of chemistry, where we'll be able to genetically encode any transformation that's compatible at least, and perform them in microbes, because I'm hoping that they will be the chemical factories of the future. And why would you want to do this? Because microbes are great at using renewable resources. Renewable resources like sunlight and carbon dioxide. And if you can imagine making what we need in our daily lives from these renewable resources using the transformations that we can genetically encode, we can start envisioning a future, a chemical future, that gets us away from fossil fuels. And so for this, I earned the greatest accolade of my life, which was to appear on the Big Bang Theory. So I don't know if you watched this television show based on students at my home institution, Caltech. I can tell you far more people watch that show than read about science or know anything about a Nobel Prize or anything else. Because over 12 seasons, though, the young stars go from being endearing grad students to being annoying professors who are lobbying for a Nobel Prize. So you may recognize some of your friends, Kip Thorne over there, and George Smoot wasted a perfectly good morning with me going over to film this show. But it has turned out to reach a lot of people. I think billions have seen this show all over the world. Now, I took on a new job almost 2 years ago now as co-chair of President Biden's Council of Advisors on Science and Technology. And I joined my colleague, Jane Lubchenco, who also works in the executive office. I got the call at a very dark time, December 2020, probably the same time you did. And we were in the deepest pit of the pandemic. Hospitals were overwhelmed. We had no vaccine, and we had very little in the way of reliable treatments. And we just finished 4 years with a president who dismissed science. And I point to the, if you see the binary code behind there, it reads hope over fear, unity over division, science over fiction, and truth over lies. And I think that well describes what science can offer in this world. Thank you.
We have time for a couple of questions.
So the silicon carbon thing, in your short life, you figured it out, but nature didn't in the entire life. Why?
Well, we gave her a new opportunity. The reagents that we use to drive this chemistry are not easily found in nature. So it's basically a new niche. And what we showed is that if you provide this new niche, innovation happens out of the diversity that already exists. The other possible reason is she could care less about silicon carbon bonds because they're not useful for survival and fitness of the organism.
Question of President von Braun.
Thank you for a great and frontiers-oriented talk. Francis, you know that many countries have written themselves bioeconomy strategies. Bioeconomy, circular bioeconomy strategies to transform our lifestyle, our economies, the way we construct buildings, run farms, and transform industries. The US has one, China has 2 months ago written itself a new one, Europe. How about sharing this science for these immensely promising opportunities around the world, in particular with low and middle income countries? How are we doing on that, and what can we do in order to enhance that?
We're still in the stage in many of the bioeconomy, the things that I've described here, where it's expensive to do it with the new technology, especially because oil has been so cheap. Now, what's going to happen is that with climate change, we're going to eventually put a cost on pumping oil out of the ground and delivering it to the atmosphere. We have to do that. And when the economics then becomes more favorable, these kinds of products diffuse rapidly across the world because they are more economically viable. And that's what's going to happen. Now, the technology is still pretty advanced, right? And so most of the startup companies are in Europe or in the United States, or a lot in China. You have to have tools.
No, okay. Once you identify this novel activity, you mentioned that you try to isolate mutants that will have that activity improved. So what kind of selective pressure do you use to isolate those mutants, and how do you identify them?
So we actually don't use selections at all, because to do a genetic selection, the function has to be tied to the fitness of the organism. Making silicon carbon bonds, no organism is going to grow on that. So we actually look at them one by one in a high throughput screening process. It very much is like breeding farm animals, right? You look at the progeny and you decide who goes on to parent the next generation. I just do that with analytical chemistry.
Ada, would you like to add something?
I want to ask. May I ask a question?
Yes? Go.
May
Okay.
I ask?
Yes. Okay. So
Can you
we move on.
tell us? Can you tell us why nature selected, or why nature is using this type of reactions and not another type? Why there are so many types of reaction that we didn't see in nature?
How many types of reactions do we see in nature?
I didn't ask how many. I just asked why that there are so many that nature does not use.
Because the opportunities are not there, or she doesn't care. Those are two
Yeah,
good,
but why?
right? The opportunities have to be there. The reagents that lead to the products have to be there, or the product has to be useful for the organism that makes it. So there are many reasons why, or we haven't found it yet. That's the other main reason. We've just barely scratched the surface of the chemistry of the existing biological world, much less the chemistry of the potential biological world. It's a wide open space.